Hi children, I'm medium sized, having curly hair, Beatrice complexion, my iris is dark, etc. etc. Why I'm describing this? This is my phenotype or the physical features which I have genetically inherited. We know in genetics we have learned that the phenotype is determined by the genotype or the genetic makeup. The genetic makeup lies in the DNA sequence in the nucleus of the cell. So what am I is all in the DNA sequence in the cell. Today I am taking a topic which will unlock the branch of biology called bioinformatics. Bioinformatics is a newly budding branch which deals with the use of computer in sequencing the genome of an organism. Today's topic is a human genome project which is associated with bioinformatics. In the coming years and in the future, there's a lot of scope in this branch. It will solve lots of problems in agriculture, industry, healthcare, environment remediation, etc. etc. This topic, human genome project, in a nutshell is the human genome was decoded and the blueprint of mankind was finally disclosed or revealed. It was a gift to the millennium, that was in 2000, but it was completed in 2003. The idea was picked up in 1984 by few scientists headed by James Watson, the person behind describing the double helix. And later in 1990, at a historic event in the White House, it was declared launched by the then President of the United States, Bill Clinton, as well as the Prime Minister of the United Kingdom's Tony Blair. It took 13 years and the project was declared completed in 2003, though the first chromosome was completed only in 2006 May. If two persons are different, definitely the DNA sequence will be different at some places. These assumptions led to the quest of finding out the complete human genome, along with the advancements in genetic engineering, computer technology, genomics, and the valuable contributions of eminent scientists from various disciplines like chemistry, physics, computer science, etc. and of course biology. This project, the Human Genome Project, was materialized and this ambitious project was very successful. So let's go into the details of the Human Genome Project. As I told you, the Human Genome Project was an international scientific research project to determine the sequence of the human genome and identify all the genes that it contained. The US Human Genome Project is a 13-year project which was coordinated by the Department of Energy and National Institute of Health. This project is said to be the world's largest collaborative project, which involved the participation of 32 scientists from over 20 countries all over the world. The team was led by Dr. Francis Collins, an American genetic scientist. This project is called a mega project. Let's see why it is called a mega project. The cost, first of all, the cost incurred for sequencing one base pair was $3 and we are all aware the total number of bases in the human genome was 3 billion. Therefore, the total estimated cost came up to $9 billion. So it was a very expensive project. So one reason why it is justified as a mega project is because it was very expensive. I repeat, The cost of sequencing one base pair was $3. Therefore, the cost of sequencing the entire bases of 3 billion, it was 3 into 3 billion, which came up to $9 billion. Second reason is the data was enormous. If we were to store these data, which is in the form of A, T, G and Cs, you know, adenine, thymine, guanine and cytosine, if it were to be stored in books, it required 3,300 books and each book of 1,000 pages and each page comprising 1,000 letters, that is A, T, G and Cs, and that too in the typed form, not handwritten, typed form. So, the space will be a little more concise. So, the data was enormous. So, it is compared to the books in the library. How many books are required? 3,300 books and each book comprising 1,000 pages and each page comprising 1,000 letters in the typed form. That much enormous was the data. So, you understood the magnitude with respect to the expense and with respect to the enormity, that is the volume of the data. So, it is easy for you to remember because I projected the 2 E's. So, if in the CBSE he was asked why is the Human Genome Project called a mega project, which is a frequently asked question, remember the 2 E's. One, it was very expensive and illustrate, and 2, it is very enormous and substantiated with the books. Clear? And because of this enormous data, it required high speed computational devices. And the purpose was to store the data, retrieve at any time and analyze. It would have not been possible if it was put in books. So, they had to put it in computers which were high speed. So, it was stored, this enormous data was stored in high speed computational devices. So, the data, the information could be stored, anytime retrieved and analyzed. Clear? So, once again I am switching over to the next slide, but before that we described the Human Genome Project as a mega project. The reason is the 2 E's. It was very expensive. The cost of sequencing one base pair or one nucleotide was $3. So, therefore, the cost of sequencing the entire genome, which comprised 3 billion base pairs, came up to $9 billion, very expensive. Then the data was very enormous. If it were to be compiled in books, it required how many books? 3,300 books, of each book comprising 1,000 pages and each page having 1,000 letters. Letters here ATGCs, the bases. And therefore, it necessitated high-speed computational devices that could store the data, retrieve, and we could analyze the data. The goals of the project. Now, let us analyze the goals of the project. The project was done with some goals. The first goal, to identify and map all the 20 to 25,000 genes that was finally revealed when the human genome was decoded. Next, to determine the sequence of the entire 3 billion chemical base that make up the DNA. So, we say that 3 billion bases or nucleotides makes the DNA. First of all, to determine its sequence, then to store these information in databases, to discover more efficient technologies with the databases, next to allow the private sector to assess the information and technologies that arise from this project, also to sequence the genomes of other organisms, and by now we have other organisms that are sequenced, like mouse is already sequenced, Drosophila, rice, bacteria, fungus and some free living pathogens in the soil. So, there are now many organisms whose genomes are sequenced. And finally, definitely there will be some ethical, legal and social issues arising from this project. So, to address these issues. It can be ethical, legal or social. It's called LC, to address LC that arises from this project. Definitely there were oppositions in this project, like cloning was opposed. Here there was a lot of opposition, taking the legal aspect, ethical aspect and social aspects. So these are the goals of the Human Genome Project. Correctly it's given in the textbook. Follow the goals correctly, learn it correctly, don't make it too much in your own words, the meaning gets diluted. So there were several goals for the Human Genome Project. Next, finally when the genome was decoded and the entire sequence was recorded in computers, what and all was revealed? The first and foremost revelation was the human genome comprised 3,164.7 million bases. It's not a round figure of 3 billion bases as always we say. Till now I was saying 3 billion bases. No, it's not correctly a round figure that is there in our DNA. It was 3.1 billion bases. So the first feature that was revealed when the DNA was decoded. Second feature is the size on an average, the gene. On an average, there were some genes which were very large and some genes which were very small, but on an average, the gene contains how many bases? 3,000 bases. As I told you, the genes, most of them were having this 3,000 bases on an average, but there were some genes which were very lengthy and some genes which were very short. The largest human gene was dystrophin, which had 2.4 million bases. See the number of bases that constitute dystrophin. 2.4 million bases constitute the gene dystrophin. Fourth, the total number of genes that were discovered were 30,000. Earlier we were guessing it would be 1 lakh genes or 80,000 or so genes, but it was found only there were 30,000 genes, and the functions of nearly 50% is yet to be known. So there's scope for research in this area. Of the 30,000 genes discovered or revealed, only the functions of 50% of the genes are known. The remaining 50% is yet to be discovered. So there's a scope for PhD or research work. Fifth, repeated sequences makes a large portion. I'm underlining the statement. This is the very important statement. There were repeated sequences which make a large portion of the DNA. Repeated are stretches of DNA with the same base composition that are repeated many times. For example, if a repeated DNA sequence is GGCCTT, it was repeated many a times. So there were repeated sequences which makes up a large portion of DNA, and this is the underlining principle or the basis of the next topic which we will be taking is DNA fingerprinting. So it is based on this aspect of the human genome, that is repeated sequences make a large portion of the DNA. And not only that, these repeated sequences do not code for proteins. So the fifth point is going to have connection with respect to DNA fingerprinting. Then sixth, of the genes, we said 30,000 genes were discovered or revealed, only 2% codes for proteins. So the remaining 98% do not code for proteins. So don't think that is junk. Very, very important. It throws light in to learn the structure of the chromosomes and to do a lot of investigation. The 98% that do not code for proteins. So the genome, the whole stretch of the DNA, only 2%, nearly 2% code for proteins. The remaining do not code for proteins. Okay, they throw light in various fields and we can research, and there is importance for that particular 98%, the existence of those 98% of genome that do not code for proteins. So remember, only 2% of the human genome codes for proteins. Seventh feature. Seventh feature is also very, very important, like the fifth feature. No two individuals have the same DNA sequence, which means that nearly almost 99.9% of the DNA sequence is similar in all individuals. We all human beings have nearly 99.9% of our genes, genome very much similar. Only the dissimilarity is just with respect to 0.1%. So we differ from each other in just 0.1%. And where this 0.1% lies? This 0.1% is the difference in the single DNA bases, just differences in a single DNA base. And how many times these occur? The locations are 1.5 million locations we see this. So once again I am reiterating the 7th point. 1.4 million locations. So remember, it's with respect to locations. There are 1.4 million locations on the stretch of DNA where there are SNPs. What are SNPs? S stands for single, N stands for nucleotide, and P stands for polymorphism. What is polymorphism? Polymorphism means differences. So there are single nucleotide differences just with respect to one base. At how many locations? Look at the number of locations. The difference is shown at 1.4 million locations. See, if I and another person have the same DNA. DNA means definitely we all have the similar DNA sequence is 9.9%. It is same, confirmed. Only that 0.1% this difference. That 0.1% difference can be illustrated by this, the three DNA shown here. See, they have depicted the cartoons of three men, one in violet, blue and yellow. Three men, let it be A, B and C. Look at the DNA sequence in all, see GCA, GCA, see GCA common, then next CGTTTAGA, CGGTTAGA, then CGTTAGA, so much similar, but look at the difference, where it became different. See, only in a single base. Only in a single base it became different. Look at the man A became different from man B just because of a single base. Look at the single base here projected as A. Whereas this man is having a single base difference is a G. Otherwise the genome of these two persons are very much identical. But you see, it differed in just one base. That one base is called SNP. I have highlighted SNP. What is SNP? Single nucleotide polymorphism, or single nucleotide difference, or single base difference. So the first man, second man and third man, the DNA is very much alike. See the sequence. For these three men is GCA, then CGTTAGA, but look in the fourth location. Look at the location, the fourth location, there is a difference. In the first man it's A, in the second man is G, in the third man it is C. So you can see that way. I showed you only with one location. I displayed only SNP in one location. That way, how many locations where you see SNPs? You see in 1.4 million locations SNPs are there, and which shows the difference between you and me, or shows the difference between two different individuals. So I told you, two individuals or almost 99.9% of our DNA is exactly similar. The 0.1 is different, and the 0.1, the difference is shown in the single nucleotide basis, which is located at 1.4 million places. Clear? So once again, this is very important, the salient features. Now to add on to this, see the 8th and 9th point I am putting here, 8th and 9th, with another color I am projecting 8th and 9th. We must know which is the chromosome which has maximum number of genes and the chromosome which has the least number of genes. It's easy to remember. Chromosome 1 has the maximum number of genes. Maximum number of genes. Now what I am considering is genes, not bases. It has 2, 9, 6, 8 genes. And the chromosome which has the least number of genes is chromosome Y. It has just 231 genes. So The 8th and 9th deals with the chromosome which has the maximum number of genes and the chromosome which has the least or the fewest genes. The chromosome which has the maximum number of genes is chromosome 1. It has how many genes? 2968 genes, whereas chromosome Y has just 231 genes. This is the reason why this chromosome 1 took a lot of time. Only by 2006, chromosome 1, the sequence was completed. I told you the project was declared completed in 2003, took 13 years, but there was a spill in the completion. This chromosome 1, which had a maximum number of genes, took another 3 more years. So by 2006 only, the chromosome 1 was completely sequenced. And because it comprised how many genes? 2,968 genes. And why? With the fewest genes. So once again, reiterating the salient features of the Human Genome Project. It's very, very important. So once again, reiterating the salient features, the first feature the Human Genome Project revealed was the number of bases. It was not the round figure 3 billion, it was 3.1 billion, or you can say it in terms of million, 3,164.7 million bases. Then second, the size of the gene on an average was 3,000 bases. However, there was the largest gene called dystrophin which had 2.5 million bases. Fourth, the total number of genes. Earlier it was estimated to be in the range 80,000 to 1 lakh, but it was found only there were 30,000 genes. And remember, of these 30,000 genes, only 50% of their function is discovered. The remaining 50% of their function is yet to be discovered. So I told you, as I told you in the beginning, the scope for research. Fifth, repeated sequence make a large portion of the DNA, which is very, very important. It forms the basis or the underlying principle of DNA fingerprinting. Then sixth, only 2% of the human genome codes for protein, remaining 98% do not code for protein. Seventh, individuals have nearly 99.9% DNA very, very much similar. Almost 99.9 is very much alike. Only 0.1% that there's difference, and that difference will be with respect to just a single nucleotide base or just due to a single base pair, and those regions are called SNPs, and it is pronounced as SNIPs. When the scientists say, they say SNPs. I is given importance to sound to make a completion in the sound, but there's no I, but it's pronounced as SNPs. There are how many such SNPs? 1.5 at how many locations there are SNPs? At 1.5 million locations there are these SNPs, and I hope you have understood what are these SNPs. Single nucleotide polymorphism or single nucleotide difference. And I have illustrated with these 3 cartoons A, B and C. Look, at only one place here they are showing difference. In the 4th base they are showing difference. Otherwise it was completely similar or replicas. But it's not one place they are going to be different. At 1.4 million locations they are going to differ. They show this SNPs. And 8th point, remember, the chromosome which has the largest number of genes is chromosome 1 with 2,968 genes, and that's why it took a longer time to be sequenced. And the chromosome with the least number of genes is the chromosome Y. Remember, boys, you have the Y chromosome. They have only 231 genes. Very, very important, the Human Genome Project, the salient features of the genome. Next. Now the approaches. How was this human gene taken out from the cell and sequenced? There's a lot of steps, and this, if you are interested, you can continue or pursue your studies in bioinformatics, genetic engineering, etc., even forensic sciences. Okay, but we have to learn superficially the approaches towards this methodology. The methodology was the same, but the approach was different, because this Human Genome Project was a government undertaking in collaboration with the other governments of the world. I told you there were many countries that participated and the countries are mainly USA and UK along with Japan, Germany, France, Italy, etc., etc. There were 20 nations involved. So it was governmental, that is called the HGP. Simultaneously, when this was progressing, when the human genome was being decoded and was announced in 1990, another company, private company, biotechnology company by the name Celera, they also came up with the same project, and they challenged to accomplish this project at a faster rate, faster than the human genome project. They challenged that they will complete it within 3 years, and also with not so much expense, with a fraction of the expense. So they accomplished it, but finally I'll show you with the help of a video. You can see how finally both of them will accomplish the project at almost the same time. So the Celera is a private biotechnology company in the USA. They also came out with a project. The methodology was the same, but the approach, the strategy was different. Both Human Genome Project and Celera, the approach was different, but the steps on methodology was the same. So let's see the two approaches. The first approach, or the express sequence tags, which was adopted by HGP, was they just sequenced only the genes that were expressed as RNA. You all know about the DNA. If I show you in the form of gene expression, this is the DNA, and from the DNA we get the RNA. Okay, and only certain sequences, we know only certain sequences do code for RNA. So just imagine only these three portions. Got transcribed into the RNA. Okay. The first approach, called express sequence tag, only sequenced these regions that were expressed. Only these regions which were expressed into RNA were sequenced, not the other intervening regions. See the other intervening region, see this one, which did not express into proteins, was not sequenced. So they didn't sequence the entire genome. They sequenced only those fragments that was transcribed into RNA. Once again, if I draw the DNA, it's not the entire DNA that is going to be transcribed into RNA. There are only certain regions that are going to be transcribed. Say for example, this region is going to be transcribed into mRNA, this region is going to be transcribed into an mRNA, and let this region also be transcribed to an mRNA. So you can see in this picture, there are some regions. See, this region is not transcribed and this region is not transcribed. So the region which were not transcribed, they did not sequence. They did not waste time to sequence this. They only sequenced the region that were transcribed into RNA. So which they sequenced? Only this and this and this. Okay, and when they sequence, they found, for example, they found, oh, this is AA, CC, GG or whatever, then CC, GG, AA and CGA, whatever. Okay, so they've only found the sequence of those fragments that were transcribed into RNA. The intervening fragments which do not code for proteins or which do not transcribe for mRNA were not at all sequenced. That was the approach taken by the HGP. Now, the second approach, sequence annotation by Celera, here what did they do is, if I draw the same thing, this is the DNA, only this transcribed into mRNA, this transcribed into mRNA, and some did not transcribe. They did not, it was a blind approach. They did not look into that and all. The entire DNA, the full DNA, the entire genome was sequenced. Whether it codes or do not codes, was fully sequenced. So the entire genome was sequenced. The approach adopted by Celera was sequence annotation, where the entire stretch of DNA was sequenced, whether it codes for protein or do not code for protein. They took a blind approach. Clear? So once again, the two approaches. There were the two approaches. One is the expressed sequence tag that was adopted by the government project called HGP, where only the expressed regions of the genes that were expressed, we know what is it expressed to, RNA, were only sequenced. That means only their nucleotides were revealed or decoded. Whereas the one adopted by Celera, the private biotechnology company, they took a blind approach. They sequenced the entire genome. Whether it codes for protein or do not code for protein, they did the entire sequencing of the entire genome, irrespective whether it codes or do not code for proteins. I hope you have understood the two approaches. But the methodology is the same. The steps are the same. First step, isolation of the DNA. First, from the sample, from the blood sample, the DNA was isolated. This we will be learning in biotechnology. Not only that, we are having practicals also related to isolating of the DNA. So, it will be more clear when we do and study. So, we are going to put hands-on practicals where we will study how we are going to isolate the DNA from coconut kernel or banana or pumpkin, etc. So, isolation of DNA, the concept will be very clear. And the second step was fragmentation with a restriction enzyme. RE stands for restriction enzyme. Again, this also is a step which we are going to learn in biotechnology in detail, but we are not going to have practicals. That when you pursue your studies in biotechnology, you will be using this particular enzyme called restriction enzyme or restriction endonuclease, and then the DNA fragments, DNA can be cut into fragments. The entire stretch of DNA can be cut into fragments. Therefore, there is a nickname for restriction enzyme. It is called the molecular scissors in biotechnology. Again, the third step also we are going to learn in biotechnology, that is amplification. Amplification or cloning in a suitable host using suitable vector. As of now, just remember the step. So, what is done is these fragments which have been subjected to cutting, these fragments are then cloned. Copies are made. Multiple copies of these fragments are made by cloning in suitable host using suitable vector. Again, this also in biotechnology it will be very clear. Now, preferably we make use of BAC. BAC is, in caps lock you have to write bacterial artificial chromosome in bacteria, or we can use YAC, yeast artificial chromosome in yeast. So, to represent this symbolically, if this is bacteria, this is the bacterial artificial chromosome. This is bacterial artificial chromosome and the host is bacteria. Similarly, if it is yeast, this is the host yeast, and here there will be an artificial chromosome like a plasmid. Why I am drawing circular, it should be circular. It is an artificial chromosome and it is called YAC, yeast artificial chromosome. So, this symbolic representation should be in your mind, because there is a property for these plasmids. They multiply by self, they are autonomous and they multiply fast. So, that vector, this is called the vector. YAC is called the vector and BAC is called the vector, and in which they are present is called the host. So, yeast is the host and bacteria is the host. So, when we incorporate, if suppose the fragment is this green color, I incorporated here. It is incorporated into the vector. See, incorporated into the vector. So, you can see a bit of green and a bit of red, mostly of red. And what happens? It is put into the yeast and it multiplies. The yeast multiplies and also this YAC also multiplies. So, the purpose of the third step is like a photostat machine to get multiple copies of the green fragment or the gene of interest. So, the purpose of the third step is to get multiple copies, and when we get multiple copies we can go ahead with the fourth step, that is sequencing, and further with ease. You need not go for a trial and error and you do not have specimen, you take many copies of the gene of interest, so you can go further with ease. So, the purpose of amplification or cloning in biotechnology is to do the following experiments at ease. we can go for trial and error methods a number of times because they multiply to thousands to 10,000 times. So, you have this gene of interest multiplied and stored. So, you can do and try, make errors, try and make errors, try and make errors. So, that is the advantage of the third one. Even in biotechnology, we are learning why we are making use of cloning. Cloning is very, very important because we can at any time make errors. Again, we can reuse, not reuse with the same gene, the gene is lost because we have made many copies, just like filling your application. When you fill an application form which is very costly, what we do, we take a copy of the application form and then you write a rough draft in the first copy. Only if it is correct you transfer into the final because it is very expensive. But if the application form is just 2 rupees or 1 rupee, you just do it directly. Okay, but this is very expensive. So, we make multiple copies. Okay, so the first 3 steps, once again I am reiterating, the first step is isolation of DNA, we will be learning and doing. So, you need not worry in biotechnology. The second step is fragmentation, that is cutting the DNA into fragments with the help of the enzyme restriction enzyme, which is called the molecular scissors. That you will be doing biotechnology, you will be learning in biotechnology, and if you are pursuing your studies in biotechnology, further you can continue with the second and third step. Third step is cloning or amplification, like the photostat machine. The gene of interest is multiplied thousands of times with the suitable vector in a suitable host, and here the vector back in the host bacteria or yak in the host yeast. It is very easy to remember that. Now, 4th, 5th, 6th, 7th and 8th step is involving computer technology. Here they got the help of computer experts. It is not humanly done, it is done with the help of computers. But you have to just remember. Next, 4th step, as I told you, sequencing using automated DNA sequencers. And this automated DNA sequencers was developed by Frederick Sanger. Okay. Then 5th step is arranging it based on overlapping regions. Again, this is not humanly done with the help of computers, like a jigsaw puzzle, how you solve it, the overlapping edges only you get it fixed back and you get the complete picture. So, here also it is arranged back, it is cut, no, it should be rearranged and brought back in a single fragment. So, that is done by overlapping regions, again computerized, again aligning it using specialized computer based programs, and again you are aligning it using specialized computer based programs. Then annotating it, that is defining their functions, again it is computer based, and finally you are assigning it to each chromosome. We have 23 pairs of chromosomes, finally we are assigning it to the respective chromosomes. So, this 4, 5, 6, 7 and 8 you will only understand if you pursue your studies in bioinformatics, genetic engineering or forensic sciences. Only thing we have to learn superficially what is the step, what just happens in each step. Okay. And with the help of a video, a cartoon video, you can see these steps once again. So, once again the methodology by both the approaches, whether it is Celera or the Human Genome Project, the first step is isolation of the DNA and the sample is blood. It can be blood or any secretion, body secretion. Mainly for this HGP, they took the blood of the volunteers. Then the fragmentation, that is the DNA was fragmented using restriction enzyme. Then third step, again in biotechnology we will be learning cloning it in suitable vectors within suitable host. Here the back in bacteria or yak in yeast. The first 3 steps once again we will be learning in biotechnology, need not worry. 4 to 8 just learn what is given in this line. Sequencing using automated DNA sequencers, which was developed by Frederick Sanger. 5th step, they were arranged based on overlapping regions. 6th, aligning it using specialized computer based programs. 7th, annotating, that is defining each one's functions, and finally assigning the genes or fragments to each chromosome. So, we will have a small look into an animated version of a video where you will be seeing the approaches by the HGP and Celera, the biotechnology company, and how the two accomplished it approximately at the same time. So, let us have a look.
The world's largest biological collaboration to accomplish this mission. The Human Genome Project proposed to sequence the entire human genome over 15 years with $3 billion in public funds. Then, 7 years before its scheduled completion, a private company called Celera announced that they could accomplish the same goal in just 3 years and at a fraction of the cost. The two camps discussed a joint venture, but talks quickly fell apart as disagreements arose over legal and ethical issues of genetic property, and so the race began. Though both teams used the same technology to sequence the entire human genome, it was their strategies that made all the difference. Their paths diverged in the most critical of steps. The first one. In the Human Genome Project's approach, the genome was first divided into smaller, more manageable chunks, about 150,000 base pairs long, that overlapped each other a little bit on both ends. Each of these fragments of DNA was inserted inside a bacterial artificial chromosome, where they were cloned and fingerprinted. The fingerprints showed scientists where the fragments overlapped without knowing the actual sequence. Using the overlapping bits as a guide, the researchers marked each fragment's place in the genome to create a contiguous map, a process that took about 6 years. The cloned fragments were sequenced in labs around the world, following one of the project's two major principles, that collaboration on our shared heritage was open to all nations. In each case, the fragments were arbitrarily broken up into smaller overlapping pieces, about 1,000 base pairs long. Then, using a technology called the Sanger method, each piece was sequenced letter by letter. This rigorous map-based approach, called hierarchical shotgun sequencing, minimized the risk of misassembly, a huge hazard of sequencing genomes with many repetitive portions, like the human genome. The consortium's better safe than sorry approach contrasted starkly with Celera's strategy, called whole genome shotgun sequencing. It hinged on skipping the mapping phase entirely, a faster though foolhardy approach according to some. The entire genome was directly chopped up into a giant heap of small overlapping bits. Once these bits were sequenced via the Sanger method, Celera would take the formidable risk of reconstructing the genome using just the overlaps. But perhaps their decision wasn't such a gamble, because guess whose freshly completed map was available online for free? The Human Genome Consortium, in accordance with the project's second major principle, which held that all of the project's data would be shared publicly within 24 hours of collection. So in 1998, scientists around the world were furiously sequencing lines of genetic code using the tried and true yet laborious Sanger method. Finally, after 3 exhausting years of continuous sequencing and assembling, the verdict was in. In February 2001, both groups simultaneously published working drafts of more than 90% of the human genome, several years ahead of the consortium's schedule. The race ended in a tie. The Human Genome Project's practice of immediately sharing its data was an unusual one. It is more typical for scientists to closely guard their data until they are able to analyze it and publish their conclusions. Instead, the Human Genome Project accelerated the pace of research and created an international collaboration on an unprecedented scale. Since then, robust investment in both the public and private sector has led to the identification of many disease-related genes and remarkable advances in sequencing technology. Today, a person's genome can be sequenced in just a few days. However, reading the genome is only the first step. We're a long way away from understanding what most of our genes do and how they're.
Okay children, you got an idea of the two approaches adopted by Celera and the government project HGP. Okay, now are you familiar with this? It is a notice you have learnt in English. This was issued by the Clinical Society, Roswell Park in March 1997, and it requested for volunteers to participate in the Human Genome Project, and I will read the notice. The goal is to decode the human hereditary information that determines all individual traits inherited from parents. The outcome of the project will have tremendous impact on future progress of medical science and lead to improved diagnosis and treatment of hereditary diseases. Volunteers will receive information about the project from the Clinical Genetics Service at Roswell Park and sign a consent form before participating. No personal information will be maintained or transferred. Volunteers will be provided a one-time donation of a small blood specimen, a small monetary reimbursement will be provided to the participant for their time and effort. So, this was a notice issued in 1997 at different points of time, because 1990 the project was launched. So, from then onwards volunteers had to donate a sample of their blood, and this was decoded, and also the content of the human genome, the blueprint, was confidential and it was not revealed to the persons who donated, the volunteers. Okay. Now, this human genome project is a stepping stone in molecular medicines. It had a very potential impact on molecular medicines. First of all, the impact was it revolutionized the ways in which diseases were diagnosed, then prognosis, treatment, prevention, and even the root cause of many diseases were investigated because of the human genome project. So, the first impact, a positive impact, was in molecular medicine. Then there was a second impact in the field of biological researches. There was a new approach to biological research. Earlier, the researchers had to study, could only study one gene or few genes at a time. But now with the entire genome sequence, they can study one gene or few gene or the entire genome. They can study for example, they can study the transcript of a tissue, of an organ, or even of a tumor, or even they can study how the different thousands of genes and proteins are working in an interconnected, coordinated manner. So, this was the impact of the human genome project, mainly in molecular medicines and also in researches in biology. And now there is scope in other fields, not only in these two areas, it also can solve problems in agriculture, in industries, in environmental remediation, energy production, etc., etc. And now many non-human models have been sequenced, and therefore there is a growing scope in this particular field. Now, I will wind up with another video, a realistic video that has documented the narratives of the eminent persons who put their head behind this project. So, let us watch.
In 1989, a group of scientists was assembled to decode the human genome under the direction of Dr. James Watson.
One of Watson's big motivations here was to try to recruit the best and brightest from many different disciplines, biology, but also physics, chemistry, engineering, because all of those would have to get together if this was going to work.
Many of the biologists, and I love them dearly, of course, were dragged into it kicking or screaming in the beginning because they failed to see the tremendous value that such an enterprise would provide the field.
The talent turned out to be all over the world. So you have to basically make it possible for those bright minds to get engaged where they are. They're not going to all move to join this effort.
Genomics has fundamentally changed the way we do science. It brought the notion of team science and collaboration to biology.
Curious thing about collaboration, it's also intensely competitive. Each person wants to achieve something personal. You know, they want to make their hit on a problem, but then immediately they want to share it with the other learns immediately. And so you ratchet each other up.
From the beginning of the Genome Project, there was a fairly strong sense of enthusiasm in the public sector, the private and academia.
We had lots of support from the public, partly because newspapers was interested in science, and I knew it. There's no partisan difference if everybody wanted to go to one for him.
I think there's a lesson here. It isn't in being a monolithic project. It wasn't being challenged, but brought people together. And I think that's what it is in the future.
When the Human Genome Project was started, it's important to note that technology of sequencing was not developed. So it's in a sense a project that was launched with a wing and a prayer.
We couldn't sequence it at that time. The machines were nothing like fast enough. There were no machines even. Sequencing was done by hand.
I thought we might have to do it brute force, and I was quite prepared to use the old
expression technology just because it was supposedly done.
There are a lot of things, that would make life easier that just hadn't been invented yet.
It'd be a 100-year project, like months working to copy a book at best. Bit by bit, we got into more and more large-scale sequencing. But the sequencing is not as important because it provides the possibility of searching by phenotype.
When I showed up at Washington University one day with a computer terminal under my arm, people looked at it and said, "What are you going to do with that?" It's a genetics department.
Genomes were supposed to be too complex to be able to reassemble them in the computer.
Without computers, we simply wouldn't get in the work.
Computers and genomics were made for each other. Genetic information is digital. It's like binary code. It just happens to use 4 digits instead of 2. It is amazingly accurate, and that's what biology gets from digital information.
The Human Genome Project contributed a cultural change about data sharing, because the Genome Project was about sharing data almost immediately. And while it seems obvious now, it wasn't obvious then. That required significant effort.
I think I'm quite lucky as a scientist because I grew up always sharing data. The community around me, the genomics community, the learning fly communities as well. It's just a default that you're going to share everything. You're going to share the code, you're going to share everything by publishing, but you're going to share full publishing, you're going to share half-baked ideas. And in fact, this is a wonderful thing, it's liberating.
It would have been virtually criminal to try to hoard discoveries that meant the difference in life and death to people.
This is information which we'll be using for centuries. The sooner we get it, and everybody has access to it who wants it, the faster we can move in terms of understanding how life works and how disease occurs.
Patients invest a certain degree of fear about genetics. Who's going to find this out? How will it be used against me?
A lot of the ethical and social considerations that we're struggling with today really have historical roots in the early 1900s in the United States, where we had many states that had laws that allowed us to forcibly sterilize people and prevent them from having children based on whether we as a society felt that they were genetically fit.
So at the beginning of the Human Genome Project, we got it right of bringing in the social scientists, the ethicists, the lawyers, and we now have helped to provide a whole generation of scientists that are focusing on the ethical and social issues of genomics.
The sequencing of the human genome was a monumental achievement. Decades of work, thousands of scientists, billions of dollars, and we get this first roadmap for what makes up the human genome. It's an incredibly exciting moment. At the same time, it's just the beginning of what's really a much longer voyage.
And let's again be audacious, but let's not be unrealistic about what's possible. I think the next 10 years are going to see some exciting developments in genomics, particularly those who start to use genomic information as part of routine medical care. And that'll be applied to dealing with diseases like cancer, hypertension, diabetes, and Alzheimer's disease.
I dream of the day when every young mother brings a young girl or young boy home from a hospital with a little gene card and says, "This is the way you should prepare this child's health care."